All right, so let's go ahead and get started today. I'd like to welcome back those of you who are returning from our session last week. I'd like to also welcome those of you who are joining for the first time to our second part of this interactive virtual mini workshop modeling COVID-19 using CompuCell3D. For those of you who are joining the first time, I'd like to begin by introducing ourselves, all of us who are helping put this on. So my name is TJ Sego. I'm a postdoctoral fellow at Indiana University, working with Professor James Glazier, who's with us today. So let's go ahead and start with Professor Glazier. If you'd like to introduce yourself, please.
Hello, I'm James Glazier. I'm a professor of Intelligent Systems Engineering here at IU. Work in the area of computational biology and multi-scale modeling. And I'm always eager to help people learn these techniques and get people started on building models of viral infection and other disease systems. Welcome.
Thank you, Professor Glazier. And how about Josh?
Good morning, everyone. I'm Joshua. I'm a PhD student at James Glazier's lab, and I'm also here to help people walk through the model and get a better understanding of how to put biological ideas into models and simulation.
Thank you, Josh. And Giuliano.
Hello, everyone. Good morning. I'm also a PhD student with James Glazier, and I'm also here to help you all with installation troubles, coding troubles, and I also work with putting biological ideas into simulation and also updating CompuCell itself.
Thank you, Giuliano. And last, Gilberto. We have Gilberto today.
Seems that he's running late. I don't see him.
Okay. Well, Professor Gilberto Thomas will be joining us shortly from Brazil. He's also here to help with both modeling aspects and computational implementations. So again, welcome. Let's get started. Today, we're diving into the code. We're going to start first by doing a quick review of what we went through in more detail in our first session last week. And again, those video recordings are available on YouTube. The links to those will be posted periodically in the chat throughout our session today. Then after that, we're going to dive in. We're going to start again with getting CC3D running on everyone's computers, whether with a local installation or using our Nanohub tool, which is our recommended medium for working today. After that, we're going to do a quick Q&A session and then take a brief intermission. The way we're doing things today is the same as our last session, so we've disabled all microphones so that we can move everyone along through this. But if you have any questions, comments, or concerns, please enter them into the chat. Professor Glazier is here to help moderate and get everyone the support that they need. As Josh and Giuliano said, they're here to help with any issues that might arise. And then after our quick break, then we're going to dive in. We're going to build some models. We're going to actually get into the code and do some actual coding, see what sorts of models we might be able to build based on the framework that we've constructed so far. And so with all that in mind, let's review very quickly. So this is a schematic representation of the modeling framework that we've built and implemented in CompuCell3D, of viral infection in an epithelial sheet. So the model consists of these basic model objects that are shown here. We have two general cell classes, or cell types, as it were, that we use to assign various model modules and properties to individual cells in simulation. So our first cell class is epithelial cells. And the epithelial cells are the cells in simulation that can actually be infected due to the virus in our simulations. These are the cells that experience viral replication inside each individual cell, and then will release virus back into the extracellular space, and can also die either due to viral activities inside the cell or various cytotoxic killing mechanisms by our second cell class, which is a generic immune cell class. Now, the immune cell class, these are cells that are recruited to our local tissue patch, and they do all of the infection fighting. So they have mechanisms of, say, chemotaxis to locate and find infected cells. They have various killing mechanisms to kill infected cells, either by contact or by secretion of an oxidating agent that will kill infected or uninfected cells at sufficient exposure. Now, with these two cell types, we then also simulate 3 diffusive fields that our 2 cell type populations will be simulated in. Now, the first one is a diffusive field that represents extracellular virus transport around the cells. So this is the extracellular virus that's actually diffusing around and can be internalized by epithelial cells to cause infection. This is also the extracellular field that's secreted into by epithelial cells when they release virus into the extracellular space. Now, the second diffusive field that's simulated is a generic cytokine field. Now, this cytokine field does a couple of different things and is primarily secreted by infected epithelial cells. Now, the cytokine field can cause activation of immune cells, and activation leads to enabling chemotaxis of immune cells, as well as secretion of our third diffusive species, which is an oxidative agent. And again, this oxidative agent is secreted by activated immune cells. And when epithelial cells are exposed to that diffusive field at a sufficiently high level, those epithelial cells, whether infected or uninfected, will die. Now, immune cells are brought into our local tissue patch in our simulation domain by a compartmental model of a lymph node. Now, the way the lymph node works is that we take a scalar mapping of our oxidative agent field. We do an integral over the field and calculate how much is in the extracellular space of the cytokine field. And then that signaling induces a pro-inflammatory state of our compartmental model, which then leads to recruitment of immune cells into our local tissue patch. And likewise, when the total cytokine field in our local tissue patch begins to decrease, we'll then find that the lymph node compartmental model will be in an anti-inflammatory state, and this will then lead to the removal of immune cells from our local tissue patch. And this is what it looks like, shown here on the right. This is a representative example of both our epithelial cells, immune cells, and our 3 diffusive species in simulation over time, moving from left to right. And this is a simulation with what we call our baseline parameter set. And our baseline parameter set was selected such that when we begin with an epithelial sheet, a 2-dimensional epithelial sheet of all uninfected cells, and we seed the center epithelial cell in the simulation domain with some virus and infect it, then over time, this will gradually lead to propagation of the infection in something like a Fisher wave that eventually leads to infection and death of all epithelial cells. So the uninfected epithelial cells are shown in blue. The green ones are infected cells, and the red ones are virus-releasing epithelial cells. The difference between these two is that infected cells are in a stage of early infection that do not release virus back into the extracellular space, whereas infected epithelial cells with more advanced infection internally will then become what we call a virus-releasing epithelial cell type that then will release virus back into the extracellular space. And then when our epithelial cells die, we show them in our sheet as being black. Now, on top of our epithelial sheet is where we place our immune cells. So our epithelial cells are frozen in that they do lots of things, but one thing they don't do is that they don't move. Whereas on top of our epithelial sheet, we place immune cells when they're recruited into our local tissue patch. And you can see this moving from left to right in this second row. We initialize our simulations with no immune cells, and due to recruitment, as you can see moving from left to right, more and more are added due to cytokine signaling by the infected cells. And those immune cells can move around. They move due to chemotaxis, due to motility, and they also contribute to a lot of the death that we see in the epithelial layer. And then you can see here the virus, cytokine, and oxidative agent fields. You particularly notice for red values, these are higher levels of the diffusive species, whichever one you may be looking at, and blue showing lower values. You'll notice a correlation between the location of significant aggregates of virus-releasing cells and significant accumulation of extracellular virus in the virus field. You'll also notice here in early stages of the cytokine field, you can see signaling by infected and virus-releasing epithelial cells. Notice this with higher levels early on. And again, this maps to the recruitment of immune cells into simulation. Should mention that this recruitment does occur due to delay to map or to model something like a distance between our compartmental model where immune cells are generated and our local tissue patch. And then lastly, you'll also notice in the oxidative agent field, you'll notice higher levels where immune cells are found. Now, some of the results that we reviewed in our last session include parameter sweeps that we did with our simulation framework in some of our preliminary analysis. And an example of this is shown on the right. So this is a parameter sweep where each plot shows a particular parameter set of interest to us with 10 simulation replicas for each parameter set. In this case, what we simulated here in our parameter sweeps was varying a parameter that changes the strength of the immune response, meaning that for the same cytokine signaling, we get either more or less immune cells, and then also varying the rate of viral internalization. So we vary a parameter along the rows, and what this amounts to is that for the same amount of exposure to extracellular virus, epithelial cells will internalize virus slower in the top row and faster in the bottom row. And for your reference, the baseline parameter set is this grid center plot in the middle of this grid. Now, one of the things that we noticed about our parameter sweeps, shown here in these shaded regions, green and reddish, is that for extreme points in our parameter space, we can find regions of certainty where we get certain outcomes. The entire system behaves mostly like a deterministic system. In this case, we're looking at the number of infected cells, and you can see here in the bottom left corner, this is for a very weak immune response and a very fast rate of internalization, and this leads to a very negative outcome. Virus infects very quickly, immune response is very slow to respond. You can see this mostly looks like a deterministic system. Now moving to the top right, we see the exact opposite. This is for a very strong immune response and for a very slow viral internalization. And in this case, measuring the number of infected cells, we see mostly none. And that's the case for all of these green shaded regions. And so what we observe with this is that in the extreme regions of the parameter space, we get deterministic behavior and certainty of outcomes. And then in between, we have something like what I like to call fuzzy boundaries between those regions, where we have lots of stochasticity, lots of uncertainty about what will happen. Now we looked at some of the causes for that variation between different simulation replicas. You can see an example of this to the right. These are 3 different simulation replicas of the same parameter set. And you'll notice here that for these 3 cases, we have very different outcomes. On the left, we have something like what we've called a positive outcome, which is that at the end of our simulation, there are lots of uninfected cells remaining, whereas in our negative outcome, there are no uninfected cells left, lots of death, and still some lingering infection. Now, the 2 primary causes that we identified in our work was considering, say, comparing the differences between these positive and intermediate outcomes. What we found was that the major cause for this difference is the stochasticity of the system concerning very early infection. In this case, the reason between getting a positive and intermediate outcome was that for this simulation replica, our first infected cell happened to die due to stochasticity of the viral replication. But the released virus was able to infect a neighbor cell, and so infection progressed. Whereas comparing the difference between intermediate and negative outcomes, what we found was that the difference between these 2 simulation replicas was not the number of cell types. We found that the first real difference between these 2 replicas was the amount of total extracellular virus in the extracellular space. And so from this, we conclude that if the number of different cell types is approximately the same, but the total extracellular virus is different, then the primary cause for this is the actual spatial distribution of infection during simulation. So from this, we concluded that stochasticity and spatial effects certainly matter when it comes to local infection for viral infection modeling. Now, one of the cases that we simulated in our early work was generating something like simulating a drug therapy. So this is shown here to the right. This is the set of equations that are used for our viral replication model. So a model of this type is assigned to every cell, and this models the various phases of viral replication inside each cell. Of particular interest is the second stage called replicating, and this models genomic replication, specifically with this term in the red box. So this is where exponential amplification of viral populations occur. Now, what we simulated was something like a drug therapy that would interfere with genomic replication during viral infection. And there's a schematic of this shown to the top right. So this is measuring this coefficient Rmax over simulation time. And the way that we simulated drug therapy by intervention, interfering with viral replication, was to say that at some point in simulation time, this coefficient Rmax is suddenly reduced from the baseline. So in this particular case, this sample, the drug therapy was applied at 12,000 minutes. And you can see that from the baseline parameter, there's a sudden drop right at the dashed line down to some reduced value. And this is simulating dosage by the magnitude of the drop, as well as the time of application of the drug therapy after infection has begun. And what we found from that in more parameter sweeps was that early treatment is better, which is no surprise. You can see this in the figure to the right. This is set up the same way. So each of these plots shows 10 simulation replicas of a particular parameter set. Now the time of application, we call it here the time delay of application. The time of application of the drug therapy for each of these simulation replicas and parameter sets is shown with the dashed line. You can see moving from left to right, that to the left we have very early treatment at the exact moment of infection, whereas to the far right we have very late infection. And then moving from bottom to top, at the very bottom row is the weakest treatment, meaning the weakest reduction in the Rmax coefficient. And then moving up to the top row, this is the strongest drug therapy, having the highest potency. And in this case, we actually simulated a complete sort of absolute intervention where the Rmax coefficient is actually multiplied by 0. These coefficients are shown here to the right. Rmax was multiplied by 0 for all of these cases, in which all genomic replication for this row stopped. Now again, we see moving or looking at the left column that early treatment is better. You can see this with the green shaded region. This is looking at the number of uninfected cells in this case. And you'll notice that in the green shaded regions that the curves don't change very much, meaning that the number of uninfected cells is approximately constant through the simulation, not much infection, not much tissue damage. Whereas moving to the right with later treatment and weaker treatment, we see lots of curves for a number of uninfected cells going to 0, meaning that everything in the very least was infected, if not killed. And this is shown in red shaded regions as well. You see this sort of similarity between this parameter sweep and the previous one. We can also identify these regions of certain outcomes, whether good outcomes to the top left or bad outcomes to the bottom right. And then again in between, we find these regions of uncertainty, these sort of fuzzy boundaries, where we can take the same parameter set, the same initial conditions, and simulate multiple times and get different outcomes. Notice this in even some simulation replicas, we get a clear bifurcation where some simulation replicas, the infection would begin to be stopped, whereas in other simulation replicas, you see a very fast decline towards complete widespread infection. And then for the clinicians, one of the things that we noticed that was particularly interesting was that for even weak potency, for lower doses, we found that for drug therapies with delayed application that even weaker doses still have a better chance of generating more positive outcomes if those treatments are applied at early times, so very soon after infection. And that's our review. That gets us to CompuCell3D. So let's begin by making sure that everyone has a working copy of CompuCell3D. We have local distributions available for people who would like to install a working copy on their local machine. I'll include these links in the chat. So you can click on those links to do your downloads in your browser of choice. If you would like to use a local installation, our preferred medium is our Nanohub tool. So this is a project tool that was deployed on Nanohub that you can launch and use in a browser, no installation required. There is a very quick registration process, but that is painless and quick. And this is also preloaded with the simulation files. So for those of you who would like to use a local installation, we'll also have to do a bit of a process to get the simulation files onto your local machine as well. For those of you who'd like to use Nanohub, everything will load and actually begin running as soon as you launch it. For anyone who has issues attempting to do a local installation, we've had some issues particularly on Macs. If it's just not happening, then we would be happy to help with support. Giuliano and I especially we would be happy to work with you after the workshop to get your local installation working. And we would just ask that if it's a matter of fighting with an installation process and missing the workshop or just launching the Nanohub tool, we would ask that you migrate over to Nanohub, and we'll work with you after the workshop. So again, there are links in the chat. For those of you doing the local installation, CompuCell3D, you'll be able to select your appropriate operating system. This will take you to SourceForge. If you do go this route, do not use the green button to download the installation. Rather, you will be able to find links in the bottom for downloading specific packages. Notice here for me, I use Windows. And so what you will want to do is use one of these links down here at the bottom. So no green button, use the appropriate download link in the bottom. Now, for those of you using the local installation, we've also made available the simulation files for the entire framework in the state that you can find in the preprint, a link for which is also in the chat. Now, I'll drop these links into the chat as well for your convenience. We make these available on GitHub. We do everything open source. And you can follow this project on GitHub with the link that was just dropped into the chat. So everything that we do is made available as we do it online. And so you can follow those links. If you use the GitHub route, for those of you who are not familiar with GitHub, you'll find a green button to the right that says clone or download. If you click on that, you'll then be presented with a blue button that says download zip. And this will download the entire repository, which is not a big package. And once you download that, you'll be able to find the simulation files a little bit buried in a terminal directory called model. Now, for those of you who are GitHub allergic, we've also made this available on Google Drive. And this monster link is also dropped into the chat. So you can follow this link. And this makes available the simulation files as well for direct download. For those of you who do go the GitHub route, you may also want to go to Google Drive momentarily. In this location, you can also find copies of the slides that we're using today, as well as the slides that we used in our last workshop, as well as the final state of all the demos that have been prepared for today. So again, those links are in the chat. Not necessary for those of you using Nanohub, though again, may want to go to the Google Drive to get your own copy of the slides for your reference and all of the simulation files at the end of each of the demos. Now, while everyone is working through getting their version of CC3D running, we'll do a quick overview of how this is actually done. So how we do model specification in CompuCell3D. So CC3D based model specification is done primarily in two different languages. The first is in what we call CC3D ML, which is XML. And in XML scripts for model specification, this is where we do basic specification and simulation details. So these are things like defining how big a lattice is for the spatial aspects of the simulation. This is specifying things like the number of simulation steps, so how long you would like your simulation to run. It's also useful for launching plugins that are built in with the libraries that do the simulating, things like simulating adhesion energy or tracking of neighbors, so on and so forth. So the general stuff is primarily done in CC3D ML, whereas the finer things are done in CC3D Python. So we do all of our fine-grained modeling in CC3D using Python programming language. And this is set up so that model specification can be done to describe events, attributes, so on and so forth, per simulation step, per cell in simulation, per lattice site in a simulation domain. So all of the finer, subtler points of doing model specification, doing implementation, as well as doing things like data analysis and real-time graphics rendering when a simulation is actually being simulated. This is all done in CC3D Python. And this is where we'll work today. We won't really get into the XML. We will be working in CC3D Python. Now, again, to review from our session last week, model specification and simulation with CompuCell is primarily done with two GUIs. The first one is CompuCell Player. And Player is the software that will load and run a simulation according to the model specification files that you load into it, as well as perform rendering of graphics in real time according to the specification, either in the preferences of Player or in the Python simulation scripts. So Player runs a simulation, it plays it. Whereas Twedit, our second GUI, Twedit is a software package that we release that is built to support model specification in CC3D. So this is a text editor that is packaged with lots of built-in support for productivity and functionality, doing common CC3D modeling tasks, and so on and so forth. So there are lots of built-in code snippets to do common tasks and model specification. We'll get into those a little bit today. We'll become more familiar with both of these GUIs as we go along today. All of our model specification will be in Twedit. All of our simulations will run in Player. Now, we spend quite a lot of time writing manuals and maintaining them to do all of this stuff. I'm going to drop links for each of these into the chat for your convenience. Very briefly, the first manual listed here is as its name. This is a scripting manual for CC3D Python. So for any questions about what's available and built in with CC3D doing model specification in Python, this is the manual to reference. The second item here, Python Quick Reference Guide. This is sort of a cheat sheet that we put together for very common tasks. So this is something to sort of keep in the background at all times when doing very basic things. And then thirdly, the CompuCell3D Reference Manual. This is documentation for CC3D ML. So this guides you through everything from setting up a basic simulation, prescribing time and lattice dimensions, so on and so forth, all the way through a detailed description of all of the plugins and built-in steppables into the libraries that you can access and use to build your particular model. So again, those links are in the chat if you want to grab those. It's always convenient to have those around. I always keep them bookmarked in my browser. Now CC3D ML looks like this to the right. Again, we won't be working in this today, but just so that it looks familiar, this is an XML specification of some system configuration items, setting up the number of simulation steps, the dimensions of a lattice. You can see that this is fairly straightforward and intuitive. Again, all of this is described in the CompuCell3D reference manual. You can see an example of specification of cell types for our actual simulation framework that we'll be messing with today. You'll notice that some of these names may look familiar. This is prescribing our immune cells and our uninfected cells. Now, more to the point of today is model specification in CC3D Python. The CC3D Python model specification is primarily done in a Python class that's called a steppable. So a steppable is a Python class that you write that gets loaded into the CC3D simulation, and the CC3D simulation will do whatever you specify in your steppable. Now, this is done in a couple of different functions. Most importantly for us today, you see on the right there are these two functions for this chemotaxis steppable. This is the steppable that implements chemotaxis by immune cells, as you see described here in this comment. The first function of interest is a function called start, and this function is called by the simulator when the simulation is being initialized. So anything that you would like to do to initialize any sort of data to make your steppable work before the simulation begins, this is all prescribed in a function called start. And our second function of interest is this function here called step. And as the name implies, this is a function that gets called by the simulator at every simulation step. So this is where the actual dynamics of the model that you're prescribing with your steppable is implemented. So at every simulation step, your steppable will have its step function called by the simulation, and the simulator will pass to your function this variable MCS. And MCS is the current simulation step number. So every time your step function is called, the simulator will always provide what the current simulation step is, and then the simulator will do whatever you tell it to in this step function. And this is where we'll primarily be working today, is building some simple basic models and describing some new dynamics, some new interactions and cellular behaviors at each simulation step. And we'll do that in our new steppable step functions. Now, if you've gotten everything installed, then as we go through this, when we load all of our simulation files, you may have noticed this if you downloaded the simulation files for local installation. The model specification for the simulation framework is actually distributed over a number of Python and XML scripts. This is specifically designed for modularity and extensibility. This is done so that building new things into this framework is easy and can be done in new simulation scripts or by doing modifications to isolated simulation files. You'll see these two files here are highlighted blue. Our Python script called viral infection VTM steppables. This is the script that defines all of the steppables for the simulation framework. And again, this is where we'll be doing our work, is with either modifying existing steppables or building new ones, and we will do all of that in viral infection VTM steppables.py. And then secondly, all model input parameters are defined in an additional Python script called viral infection VTM model inputs.py. This is the script that we messed with a little bit last week in our first session. So when we change actual model parameters or simulation specific preferences that are defined in Python, which we'll do a little bit today, we'll change those values and variables in this model inputs script, viral infection VTM model inputs.py. Now, when we get into the simulation script defining all the steppables, what we'll find is just over 1,000 lines of Python script that are defining quite an array of different steppables. And we will not talk through all of these. Some of them aren't particularly interesting for what we'll do today. But one thing that you will notice is a certain methodology for model specification that we have implemented with this framework and that we encourage when it's further developed and extended. As the names imply, most of these steppables implement specific modules in the framework. So you'll notice here to the right, for example, we've talked a little bit about things like internalization and replication. You'll notice that for this module, viral internalization, this is a separate module in the framework. And this is implemented in a corresponding steppable called viral internalization steppable. And most of these implement one of these particular modules. There are a couple of sort of supporting steppables like cell initializer steppable. This defines the initial simulation configuration. Sim data steppable. This does lots of data analysis and some of the real-time graphics reporting of data. But most of these will implement specific modules. When we do some of our model building today, we will make some new steppables, and those new steppables will implement specific new simple models. The CC3D Python has lots and lots of details, lots of special attributes, lots of built-in functionality to do things very quickly to make model specification as simple as possible computationally. We'll just go through a few of these that are relevant to today. For every steppable that we work with, there are lots of attributes that can be accessed by typing a command self dot and then some attribute or some function that's defined for every steppable. An example that we'll work with today is doing things like accessing the cell types that have been defined for a particular simulation. So we notice in the CC3D ML specification in the example a few previous slides, there was a cell type defined as uninfected. We do that in CC3D ML, but if we go to CC3D Python and would like to reference that cell type and how it's defined in implementation computationally, in any steppable, we find that we can just type self dot and then in all caps uninfected. And variables like this, attributes like this will be defined for every cell type for a particular simulation. Some model specification is done on the basis of cells, some is done on the basis of cell types. And when doing references for model specification based on cell type, you can always expect that a steppable will have specification according to this convention, where the IDs that are assigned for particular cell types will return or be returned when using this sort of convention. If we get far enough along today, we'll also find that we can do things like accessing the value of diffusive fields at specific locations in a simulation domain. So for example, we've talked about our extracellular virus diffusive field. If we were, say, interested in Python to look at a particular value in the extracellular virus field at a location with coordinates X, Y, and Z, in Python we can very quickly access that value in any steppable with just the entry self.field.virus XYZ, where the name of the field that's defined in the CC3D ML is then the corresponding attribute for access in Python. There are a lot of utilities that are also defined for the steppables. We'll use a few of these today. These are well described in the Python reference manual, but just a couple of examples to generate and do a loop over all of the cells that are currently available in simulation in Python. You can just very quickly type for cell in self.celllist. Cell list is an inventory of all of the current cells in simulation. Another example that we will use today is doing things like generating a list of current cells of a particular type. So another cell type that was prescribed in our CC3D ML example was the infected type. And you'll notice here is this example to the right, to generate a list of all the current cells of the type infected. In Python steppables, it's as simple as typing self.celllist by type and passing the cell type of interest. In this case, an infected cell type. So this will return a Python list of all of the cell objects that have this particular cell type. Now, we've also set things up so that information can be passed between steppables. So we do lots of connectivity. Internalization, for example, leads to viral replication. Replication leads to viral release, so on and so forth. So to do these sorts of interactions between stepables and also passing information, there's a global Python dictionary that's accessible by every stepable that's defined in a CC3D simulation. And that global Python dictionary is called Shared Stepable Vars. So any Python stepable in CC3D has this attribute. And when performing manipulations on that dictionary, those manipulations are reflected to every other stepable. This is, again, nice for doing things like connecting stepables that interact or lead to events in other stepables. We also use it, for those of you who are more into some of the computationally intensive work, we also use self.shared stepable vars to do interactions with the outside world, meaning the outside world with respect to CC3D. So we have a Python API where information can be passed into and out of CC3D from Python and made directly available to the stepables in a particular simulation. That's, again, done with self.shared stepable vars. Now, lastly, all of this functionality is defined on a class, a Python class that that all of our stepables will inherit from. We'll see this when we actually get into the code. We'll do some minor changes concerning this, but as we get into the stepables, we'll notice that all of the stepables that are defined will inherit from something like a Python class called stepable base Py. So all of these features are defined on those classes. For the framework that we've been developing for this particular project, we've been developing a specialized base stepable class. This is an extension of stepable base Py. You'll notice this when we actually dig into the code. You notice that most of the stepables will inherit from this Python class called viral infection VTM stepable base Py. So in addition to all of these features that are defined for general CC3D stepables, we also have additional features that are defined on this base Python class. We'll use some of those today. Now, each cell in simulation, the Python cell objects, these also have attributes that are defined and updated by the simulation, or by the simulator, rather. So when we work with specific cell objects, we'll find that we have convenience attributes, something like, for example, cell.type. So just as we were talking in the stepables about having easy access to the cell types that are defined in a simulation by self.say infected, when we handle a specific cell object, we can find that cell.type will also reflect the integer of the cell type for this particular cell. So for example, if we wanted to, say, change the type of a particular cell to infected, in a CC3D Python stepable, it's as simple as just saying that cell.type equals self.infected. And this quite literally just changes the actual cell type of this cell instance to the infected type. We also have lots of attributes that are updated in simulation based on the state of the cell concerning its location. So for example, we can very easily get the center of mass of a cell in simulation using these attributes XCOM, YCOM, and ZCOM. Every CC3D cell in simulation is also given its own unique Python dictionary. The attribute is dict, cell.dict. And this is ideal for defining things like custom cell properties. In the case of our framework that we've been building and are working with today, we use the dictionary assigned to each cell to keep track of the number of cell receptors that are available for viral internalization. In our implementation, you'll find that the line is cell.dict with the key receptors. And then lastly, we can also attach ordinary differential equation solvers to individual cells. We talked earlier about the viral replication model, and that's modeled with a set of ordinary differential equations. Each cell is assigned an instance of a solver that will solve whatever ODE system that you've assigned to it. That solver can be accessed for each particular cell with cell.sbml, as shown here. Now, model specification for these ordinary differential equations in CompuCell can be done either in SBML, in antimony, or in CellML. This is documented in the Python scripting manual, so you can see more details about that. We won't get into this too much today, but if you actually dive into the simulation scripts for the framework, you can find lots of instances of using antimony to implement the viral replication model and the immune or the lymph node compartmental model. Excuse me. And in CC3D Python stepables, there's just a very basic convenience function here defined for the stepables. It's called timestep SBML, and this just tells the CC3D simulator to integrate in time every ODE solver that's been defined, whether by cell or in general. So this is an example of this, again, shown here in the bottom right. This is the viral replication model. Again, this is modeled with 4 ordinary differential equations. And up here to the right, the top right, this is an example of the spatial simulation in the epithelial sheet. You can see that there are some infected and virus releasing cells. There's one dead cell and lots of uninfected cells. Now, if we actually go and look inside any of these individual cells, we find that each of them has an instance of the viral replication model, as well as instances of internalization and release. Now, the cell.sbml solver that's attached to each cell can then be used to access the instance of the viral replication model for the particular cell. And in this case, for the simulation framework, the name of the viral replication model is shown here to the right. So when handling a particular cell in CC3D Python, we can access the replication model for that cell using cell.sbml.viralReplication. And now every variable and coefficient that's defined for that particular cell and model can be accessed using keywords that correspond to the names of the variables in the model specification. So in this case, shown here in the bottom right, in our drug therapy simulations, we did some changes in real time of the coefficient Rmax in the model specification for this system of ordinary differential equations. Rmax is defined as replicating rate. Now, programmatically, at any time in simulation, we can then access that value and change it using this line shown in the bottom center. You can see this is for a particular cell, accessing the SBML solver, then the viral replication model for that cell, and then the name of that coefficient that we're interested in, which is replicating rate. So we'll do this a little bit today. This is how this is done on a cellular basis. Now, hopefully, everyone by now has either launched their Nanohub tool or has gotten their local installation downloaded and installed. So let's do this. I'm going to launch my own version of CompuCell. I'll do this both locally and in Nanohub to start. Once we actually get to writing some demos, I'll work locally on my machine, but again, you can do all of this in your own Nanohub tool. So we will start by launching player. So you should be able to just go to something like a start menu and type CompuCell3D, and you should get a shortcut to launch player. And when you do, it should look something like what's on my screen now. So this is what CC3D player looks like. I have a workspace here. This is where lots of different graphics rendering screens will go. We'll get some feedback from the bottom, and then we have a model editor to the left. Now, I'll do this first for the local installation, and then I'll switch over to Nanohub and do this. Let's work through actually loading the simulation files into player. So we can do this by going to the top left to the menu bar, and going to File, and then Open Simulation File. So to the top left in the menu bar, go to File, and then Open Simulation File. Now when we click on this, you can navigate to wherever you downloaded the simulation scripts, and you should find a file named viral infection vtm.cc3d. So again, this is in the main directory with all of the model specification files you downloaded from either GitHub or Google Drive. You should find the script viral infection vtm.cc3d. Every CC3D project has a CC3D file, and this file specifies all of the simulation scripts that define the simulation. So in player, we can click viral infection vtm.cc3d, select it, and click Open. And if you were successful, then you should notice in the top right, you should see that it reads CompuCell3D player, and it should say viral infection vtm.cc3d. Now I'm going to switch over to Nanohub, to my Nanohub tool. For those of you who launched Nanohub, you may have found that it looks something like what I'm looking at currently. Player opens and actually begins by running the simulation. May also be somewhat disorganized. For the Nanohub users, you can modify the size of the screen to make some more space by clicking and dragging the bottom right corner of the tool for your reference. Now, your screen may look a little bit differently than mine concerning what's actually plotted. That's okay. We'll get everyone up to speed on getting their workspaces set up properly. We can change what's displayed. We can create new graphics windows. We'll go through all of that momentarily. For now, let's get everyone on Nanohub loaded up. So the Nanohub tool does launch with the simulation script loaded, but you can also open different simulation scripts from the Nanohub tool. And let's go through that very briefly. In the Nanohub version of Player, opening a simulation script is the same within Player. We go to the menu bar at the top left and go to File. Actually, make sure that our simulation is stopped first. You can stop a simulation if you refer to my screen, see where my mouse is. This fourth button in the top left, it's a button called Stop. A simulation in Player has to be stopped to be able to load a new one. So you can stop the simulation again if you refer to my screen, see where my mouse is. Once you've stopped a simulation, you can go to the menu bar, go to File, and then Open Simulation File. And when we select File, Open Simulation File, this will open a dialog. You'll notice that to the left you'll see a directory that should have your username. Mine is TJ Sego. So I'm going to select that. You can select yours. And you'll notice then that to the right in the main window, there's a directory available called CompuCell3D _demos. You can go into that by double clicking. So again, CompuCell3D demos, double click that. This will then present two more directories, the second one of which should read coronavirus. Now you can double click that to go there, which we would like to do. So double click coronavirus. And this gets us to our simulation scripts. Most notably for our intents now, we would like viral infection vtm.cc3d. Again, this is our main simulation script. This tells player what all is used to execute this particular simulation. So let's select viral infection vtm.cc3d if you're working in Nanohub. To the bottom right of the dialog, click Open. And you should see in the top left that the Nanohub deployment of player also reads viral infection vtm.cc3d. Now, the controls for player are as follows. I'll do this in Nanohub, but all of these controls in player are the same for local installation. I'd like to know how to run or pause or stop a simulation that we've loaded. So these features are available in the top left with these 4 buttons. In Nanohub, I already used the stop button. Now, just under the menu bar, you should find these 4 buttons in the top left of player. The first button, when you hover your mouse over it, it should read Run. And this will run the simulation. This will move simulation step by simulation step through the simulation all the way up to the number of simulation steps that are defined in the CC3D markup language specification. That's the first button. The second button, if you hover over it, it should read Step. And the Step button will perform 1 simulation step. And then it will pause. And you'll notice that if you don't have a simulation running, as is the current state of my player on Nanohub, nothing is running. If you don't have anything running, go ahead and click Step and see what happens. So the second button, click Step. Now, if we give player a little bit of time, mine just completed, you should notice that player refreshes everything. Should see some new graphics windows. And most importantly, when you look at the bottom left of player, you should see some red font that just says Step 0. You'll also notice in the console, you're probably getting some reporting that says Step 0 and flips and some other interesting things. We're at simulation Step 0 and paused. So in this case, the framework has now been successfully loaded. It's been initialized, and the simulation is paused. And if we click Step again, we'll get another simulation step. Now if we hit Run, the first button again, then we should see it start going. You'll see in the console, you'll see the steps counting. Depending on the preferences that your player has been configured for, your screen may be updating more or less often. My Nanohub tool launched is updating every 10 simulation steps. If I switch over to my local version, I believe right now mine is configured to update every simulation step. So if you're seeing something a little bit different from mine, that's fine. All of these configuration properties can be modified according to your preferences. So for example, let's do a little configuration for player. Let's start by pausing our simulation. I'm now in my local installation. My Nanohub tool is still running. I'm going to switch over to my local version. And in the top left, the next button after step is the button called pause. You can click that. And you'll notice after you've clicked pause, you look at the console, you see that you're no longer getting any reporting for new simulation steps. So the simulation is still loaded, and it's effectively been frozen. And if you hit step or you hit run, it will continue from the moment where you paused it. Now, if you like to see graphics updated more often, you can go in player to the menu bar. You can go to Tools and Configuration. So Tools and Configuration. And this will open up CC3D preferences for player. And you'll have some tabs where you can do all sorts of different customization. For our purposes now, let's go to the first tab that says Output. So again, the first tab says Output. And then you'll have a first entry here called Update Screen every nth MCS. And the number that you enter here will be the frequency at which all the graphics will be updated for spatial results, meaning the plotting of the cell field, the plotting of whatever diffusive fields are being simulated. All of those will be updated according to a frequency that you specify in this first entry, update screen every nth MCS. So you notice on mine, in my local installation, I have mine set to 1 currently. And when I go to my Nanohub tool, again, mine's running, so I'm going to hit pause. And then in Nanohub player, we go to Tools and Configuration. Again, you get the CC3D Preferences dialog. Same tabs, possibly different entries. And the first tab, Output, let's go there. And for some of you, this may have read 10. If you'd like more frequent updates, you can modify this here. And click OK in the bottom right of the dialog. Now I'm going to switch back over to my local installation. I'm going to do a little bit of organizing. So some of you, if you look at my screen, I currently have plotted the 2 levels of the cell field. Again, we have a 2-dimensional sheet of epithelial cells on one level, and then above it, immune cells are added on top, and those immune cells will move around on top of the epithelial sheet. If you refer to my screen, I have 2 graphics windows showing each level of the cell field. And if you don't have one of these, let's pretend now that I don't. I'm going to close my plotting of the immune cell level. So say you had a state like this, where you can only see one cell field level. I'd like to be able to see both. You can create another graphics window in Player with the last button along the ribbon at the top. You can see this on my screen. I'm hovering over the last button, and it reads New Graphics Window. Now, when you click this, again with a simulation script already loaded, I get a new graphics window. And in my case, it happened to present exactly what I was looking for, but let's say that it didn't. Let's say there's just the oxidative agent. So if I had started here, and I would like to be able to also see the immune cell level in the cell field, I can do this very easily in my new graphics window by selecting this drop-down menu and selecting a result of interest to display in this window. You see here, there are 4 results available for this framework. There's first the cell field. This is where all the cells are simulated. And then there's a separate field entry for each of our 3 diffusive fields, the extracellular virus, cytokine, and the oxidative agent. In the framework, it's just called oxidator. So to look at the cell field, let's click on cell field. And if you notice on my screen, I now have 2 plots of the epithelial sheet. Now, again, referencing the configuration of the actual simulation of the cells, we have our epithelial sheet that's in one level, and that level is in the XY plane. So we are looking in CompuCell at the XY plane of the cell field. And in this implementation, the epithelial sheet is placed at coordinate z = 0. And you can find this in all of your graphics windows that are showing various spatial results. You'll notice that each of them next to the results will also have a number. For this framework, it'll either say 0 or 1 because we have 2 z coordinates. The first coordinate, z = 0, again, is where the epithelial sheet can be found. And then at z = 1 is where the immune cells are placed. And in this case, we have a graphics window that's looking at the XY plane of the cell field. We can then change to coordinate z = 1 for our plotting by going to where we see it says 0 and just entering 1. So now if you're looking at both the epithelial sheet and the immune cells, what you're looking at is 2 cross-sections of the same cell field. In the case of looking at the epithelial sheet, you're looking at a cross-section of the cell field at z = 0 in the XY plane. If you're looking at immune cells, you're looking at a cross-section in the XY plane at z = 1. Now, just as a warning, we've had a bug that's been plaguing us in CompuCell recently where at times, at random, one of these screens may go just completely black. And if that happens, you've done nothing wrong. If you're following along watching my screen, you'll certainly see it at some point. The screen goes black, you can simply close it, just create a new graphics window, and reconfigure it to whatever it was that you were looking at. Excuse me. Now, if you're following along with me, I'll particularly be interested in looking at the cell field in the 2 levels, as well as the virus field. You have some plots that may or may not have loaded that look like the ones on my screen. those didn't load for you, that's okay. We'll configure those momentarily. More importantly for now, lastly, before we move on to Twedit, is how to organize all of this in a coherent way. So you can plot, oh, here you go. If you refer to my screen, here you can see an example of our CC3D bug that's been plaguing me for months. My virus rendering went black. That's okay. Just close that window, open another one, and go to virus, and at z equals to 0. You'll notice in the plots of the diffusive fields, you'll be able to see the outlines of the cells as well. When I look at the virus field at z equals to 0, I see a grid. Currently, it actually looks a lot like the uninfected epithelial cells, but this is in fact just an outline of the boundaries to help you get a sense of what's going on in the cell field and how those are related to what's going on in a particular diffusive field. Now, lastly, let's organize Player a little bit so you can generate all sorts of graphics windows to look at all sorts of different things. As the workspace gets more and more cluttered, it may become more and more difficult to manage all of it. So a very easy way to quickly organize things into a nice grid of plots is to go to the menu bar in Player and go to Window. And in Window, you can go to the third item, which says Tile. And you should find that when you go to Window and Tile, that whatever graphics windows you currently have open, you should arrange them in something like a nice grid of tiles so that you can easily see everything all at once. Now let's launch Twedit. So in Player, it's a very easy way to quickly launch Twedit, and that is with a button in the ribbon along the top. This is true for Nanohub and local installations. You refer to my screen, you'll find a button along the top in the ribbon that I'm hovering over. When you hover over it, you should find that it reads Start Twedit++. So in the top in the ribbon, you can find the button that I'm currently hovered over. It'll be the third button in the second section of the ribbon. Start Twedit, and let's click it. And you should find that Twedit launches with a blank document with the generic name along the top and an empty project pane to the left. Again, this is true in Nanohub as well. In Nanohub Player, there's a button for Start Twedit. Click that, and in Nanohub, Twedit will also launch with a blank document called New Document 1. You can easily switch back and forth between Player and Twedit in Nanohub by going down to the bottom. You can see that you can easily select between the two GUIs. Now let's work in Twedit here for a little bit. I'm going to do this first in Nanohub and then switch over to my local installation of Player. Now, what we would like to do is load the simulation files into Twedit so that we can edit them and see what happens in Player. So in Twedit, we can do this similarly to Player. In the case of Twedit, in the menu bar, you'll find an item, CC3D project. Again, this is true for local installations in Nanohub alike. In the menu bar, click on CC3D project. And the second item will be Open CC3D project. Let's click that. So in the menu bar, CC3D project, and then Open CC3D project. And when we select that, should get a dialog, Open CC3D file. Nanohub happens to present the simulation scripts, but let's go through this from the top for Nanohub first. This is the same as when loading from player. On the left, you'll find a directory that has your username. Mine is TJ Sego, so you can see on my screen. I have a directory with my username. Select that. And this will present the directory CompuCell3D demos. Again, we'll double click that to go into there. And in CompuCell3D demos, we find the directory coronavirus again, just as in player. So we'll double click coronavirus to go into there. And we find viral infection vtm.cc3d. So we'll select viral infection vtm.cc3d in Twedit and then click open. In the local installations, the only real difference is finding where you saved your simulation scripts. Just as in player, you have to locate where you saved the downloaded simulation scripts, but otherwise the process is the same. In the menu bar, CC3D project, open CC3D project, and navigate to wherever you saved your simulation scripts, and then viral infection vtm.cc3d, and click open. Now in the left, in the CC3D simulation items, you should notice that there is a clickable item in Twedit called viral infection vtm.cc3d. So let's double click that simulation item, viral infection vtm.cc3d, along the left in the project pane. When we double click that, should see all of the simulation scripts open in Twedit. Again, this is the same in Nanohub. Should have this clickable item on the left, viral infection vtm.cc3d. You can double click that, and that'll load all of the simulation scripts. Now, some of these should look familiar. I'm going to switch to my local installation. In particular, viral infection VTM model inputs.py, and you can read these in the tabs along the top, as well as viral infection VTM steppables.py. Now, one thing to note when doing specification in Twedit is these little icons for each of these simulation scripts. You notice that when you loaded them, all of those should be blue. But if you make an edit, I just entered a space into my viral infection VTM steppables.py, and you'll notice on my screen, or if you make some tiny modification in one of your scripts, you should notice that then the disk next to the script name should turn red. These are notifications to let you know that there are unsaved changes in this simulation script that need to be saved before they'll be reflected in player. So in my case, I hit save. You can do that in the menu under CC3D project. or using the appropriate hotkey for your operating system. In Windows, it's just Control-S. And you see that the blue disk comes back. Now, very quickly, let's do a tiny bit of setup to make sure that everyone is able to see the additional screens or windows that I have in Player on my screen. In case you didn't have windows pop up showing population data or the immune response model, like you may have noticed in mine, and you would like to have those, let's go through very quickly how to enable these windows, as well as other data reporting that we've implemented in this framework that may be of interest to you. We won't work with them much today, but there is plenty available to work with. If you go to viral infection VTM model inputs.py, so in Twedit, along the top, let's click the tab for viral infection VTM model inputs.py. And what you should find, if you scroll down, there are lots and lots of model parameters. Again, this is the script that defines all of the model inputs for the various modules that are defined in the framework currently. But at the top, beginning at, should be at line 12, should see all of these different variables with prefixes, either plot underscore or write underscore. And you should notice that there are pairs of these. On my screen, I have highlighted the pair for plot VRM data freq for frequency, FREQ, and then also write VRM data freq. Now these are pairs of preferences for various data analysis that we've configured in the framework to either plot in real time in Player or to write. And by write, I mean write to disk. So now if you happened, for example, to launch Player and start a simulation, you may have noticed that you didn't have a graphics window pop up as it did in mine that was plotting the population data. So if you recall in the list of stepables in the framework, one of them was called SimData Stepable. And one of the things the SimData Stepable does is it'll count the number of the various cell types in simulation, every simulation step. And if you ask it to plot that data using the variable at line 22, plot pop data freq, it says plot the population data frequency. You can see this described in the comments for each of these variables. If you didn't have that graphics window pop up in Player, chances are it's because your variable at line 22 was set to 0. You'll notice in mine, at line 24, my write pop data frequency was set to 0. And this is just our convention to specify that we don't want that functionality. So all of the write variables should have been set to 0, meaning that they shouldn't be actually outputting any of this data. Whereas for my case, at line 22, mine was set to 10. And this is specifying that every 10 simulation steps plot the population data in Player. Now, as a brief exercise, you go to the variable at line 22 and change it to say 1. In my case, mine was set to 10. I'm going to change it to 1. And I find that along the top in the tab, my disk for viral infection VTM model inputs.py is now turned red. Now I'm going to save to get a blue disk. That means that everything has been saved and will be reflected in Player. Now, if you make that change and make that save, then you can now go back to player. And if you have a simulation that's running or that's paused, you can hit stop. And now select run in the top left, the first button, run. This will start a new simulation. And you should find that if you didn't have a graphics window plotting the population data, you should have one now. If you did have one, but you changed the plotting data frequency, you should find that the graphics window is now reporting the population data at the new frequency that you specified. In my case, mine was set to 10. My immune response model is still reporting at a frequency of 10 simulation steps, whereas I changed my population data to 1. You can see that the reporting frequency is updated accordingly at the next launching of the simulation. And then for those of you who may not have gotten the immune response model, this is plotting the state variable for the compartmental model of the lymph node. Most importantly, the state variable S that's being plotted. If you don't have it, we'll enable that momentarily. You can refer to my screen for now. You'll notice that when this state variable S is very positive, you should then notice that the number of immune cells in the cell field will be increasing. This is indicating a very pro-inflammatory state. And when the state variable S becomes very negative, you'll then find that the number of immune cells in the simulation in the cell field will be decreasing. And that is indicating a very anti-inflammatory state. Now let's go back to Twedit. And again, there are quite a few other plots and exporting features available for some of the different data that we've set up to do the immune response model plotting. If yours didn't pop up in player and you would like for it to, you can find that variable at line 30. Plot IR data freq for plot immune response data frequency. And you can just set that to a frequency of your choice. I have mine set to 10. I'm going to set it to 1. I find it interesting to watch how that state variable S behaves and then the changes in the corresponding immune population. And now I'll save. I have all my blue disks, so everything is loaded, and we are set up and ready to go. So with all of that, we'd like to take a pause here and open it up to all of you for any questions that we might be able to answer, whether about the model or the simulation scripts. This is also sort of a last call. We'll do a break, a very brief intermission for about 10 minutes after this, in case there are any last minute issues with CC3D. So again, we're keeping microphones muted to expedite the process, keep everyone together. So if you have any questions or comments, please enter them into the chat. All of us are available to answer those questions. And then after that, we'll take a brief pause. Give it another minute or so in case anyone's working on getting any questions in. All right, and in that case, I think let's go ahead and take a brief pause. Let's let everyone stretch their legs, get some water or coffee for those of us who are still in morning hours. So let's do 5 minutes. It's currently 10:37 our time in Indiana. Let's come back now 10:38. Let's come back at around 10:43 Eastern time. So let's take 5, and then we'll come back and we'll start doing some actual work in the code. So I'll see you all in 5.
So maybe while TJ is taking a little bit of a break, I'd like to answer the question from Christian Richardson, which is how are immune cell recruitment and lymph node state determined? In reality, of course, there are a very complicated set of signals involving cytokines and other signaling molecules that travel both within the immune system and between the tissue and the immune system, both pro and anti-inflammatory cytokines and chemokines. In addition, you have antigen-presenting cells that move from the tissue to the lymph nodes and other systemic immune tissues. And you have movement of immune cells from the lymph nodes to the target tissues. In this particular model, we have a very simplified version of an immune system that's based on really a cancer-derived model, thinking about T cells. The model looks at the concentration of the single cytokine field that we have. That cytokine field is sent to the lymph node model with the delay. And as the cytokine field concentration goes up, the pro-inflammatory state of the lymph node goes up. When the pro-inflammatory state of the lymph node gets to a certain level, it creates, with the delay, a cell in the tissue, an immune cell in the tissue in an inactive state, and reduces the pro-inflammatory state. There is a positive feedback, though, because the immune cells themselves do secrete cytokine, although in the particular simulation that we're running now, the gain is fairly low. So what's happening here is that the cytokine level in the tissue, which is determined by the number of infected cells and the number of immune cells in the tissue, sends a signal to the lymph node about the net pro or anti-inflammatory state. The lymph node then generates cells, immune cells in the tissue, and reduces its inflammatory state. If the net inflammatory state is negative, the lymph node actually takes immune cells out of the tissue. There isn't an explicit lifetime for the immune cells resident in the tissue. So that's the basic immune cell model. I hope that helps. It's a caricature of the reality of the immune system. And our hope is that people like you in this call will come back and tell us how to improve that. Obvious things that we'd like to do would be to include interferon-induced antiviral resistance in the epithelial cells, include neutrophils and macrophages and phagocytosis, and to break up this single immune cell type that we have here, which is a caricature, into explicit T cells, natural killer cells, CD8 plus cells, CD4 plus cells, and other appropriate species. Also to include the explicit role of dendritic cells in antigen presentation. And we'd love to work with people who are interested in implementing any pieces of that model and telling us what you think is important in doing that. I hope that helps a little bit in answering that question.
Give it another couple seconds. Let anyone else get back who may have stepped out. All right, let's go ahead and get started into the code. So the way I'd like to start today is by building our drug therapy simulation. So this is actually not a standard feature in the simulation framework, but rather a scenario that we added in after we built the general framework. Fortunately, this can be done quite simply with just a couple of lines of code. So as an overview, or more of a synopsis of what we're going to do, we would like to simulate the event where some drug intervention inhibits genomic replication. Now again, if you refer to the bottom right of my screen, this is the set of ordinary differential equations that we use to model viral replication inside each cell, each epithelial cell in simulation. We have an uptake term that's passed from the viral internalization module. So this is a source term. During an internalization event, it starts the sequence of events of replication inside of a cell. And at the very end, find on the bottom right, there's this term release. And this value is then passed to the viral release module that then simulates release of virus back into the extracellular space. And in the viral replication model, this is the sort of throughput process of the virus cycle. In particular, the second expression, again, describing genomic replication with this term that's boxed in. This is where replication actually occurs, exponentially so. And as we discussed previously and in the last session, in our drug therapy simulation, what we say is that we begin with our baseline parameter set for this coefficient, R max, just as every other coefficient in the baseline parameter set. And then at some time in simulation, that coefficient will be reduced, and this simulates a drug intervention. So in the very simplest case, we can say that this is uniformly applied and instantaneously. And when simulating drug therapy, two particular elements of interest would be both the dosage and the timing. And so what we'll say is that our two model inputs for this particular simulation will be representing the strength of the dosage as well as the time in which it's applied. So we will specify new model inputs that will specify the time in simulation that the intervention is applied, as well as the dosage, which we will describe as a reduction coefficient from the baseline parameter set. So this is an overview of what we're going to do. We're going to add this model input. We'll call it a reduction coefficient in genomic replication. And we'll describe this as such. We'll describe the coefficient that's used to multiply the baseline parameter set, or the baseline parameter value by. So if we want to impose, say, a 10% decrease in the replicating rate, then we'll prescribe a coefficient equal to 0.9. And then we'll also prescribe the simulation step at which this reduction is applied to all epithelial cells. Now, in our case, we don't need to define a new steppable for this particular scenario. Rather, we're just going to modify an existing one to start. So again, all of the stepables are defined in the script viral infection VTM stepables.py. So if we go to Twedit, along the top, you can select viral infection VTM stepables.py to make that script available. And we can go down to around line 133. At line 133, in viral infection VTM stepables.py, we'll find that we are in a step function, which begins at line 124. And this step function is being defined for the viral replication stepable, which begins at line 105. So we're going to modify viral replication stepable inside of its step function. As discussed previously, all of these stepables will inherit from a Python class that defines all of those useful utility functions and attributes and so on and so forth. Ordinarily in CompuCell, that base class, if you refer to my screen very quickly, ordinarily that base class is called steppable base pi. Inheritance is implemented like this. In our case for this project, the special steppable base class is called viral infection VTM steppable base pi. So this inherits from the CC3D steppable base class, and it also defines some additional convenience functions and processes that are specific to this project. And you can find those definitions in the Python script viral infection VTM steppable base pi. We won't get into this today, but you can find all of that in the simulation files as well. Now, let's do our drug therapy simulation. Let's start at line 133. We should be at a comment that says do viral model. And at line 133, I'm going to begin by returning a couple of times to make a little bit of space. And let's annotate what we're going to do. After having made some space at line 133, let's then begin by adding a comment. It should show up as green. And let's just put here something like drug therapy. Now, after our comment, let's start by defining our two coefficients. I'm going to flip back to my slides very quickly. And we're going to begin by defining both our reduction coefficient in genomic replication, as well as the time of application of the drug therapy. So back in Twedit, at around line 134, let's start by defining the coefficient for reduction. Let's call it mag underscore reduce. Mag underscore reduce. This will be our reduction coefficient in the replicating rate coefficient Rmax. This is what we will multiply that baseline parameter by whenever in simulation we apply our drug therapy. Let's say that mag reduce, for now, let's just say that it's equal to 0. Now, after defining mag reduce, let's return. And at around line 135, let's add another model input, our second one for the scenario, which is the time of application. Let's call this variable time underscore reduce. So our variable for the time application of the drug therapy, we'll call it time underscore reduce. And for now, let's just say that that's equal to 50. Now for your reference, the simulations that we'll be doing today, these are for 1,000 simulation steps. And so for this current set of model inputs, for a reduction coefficient of 0 and a time of application of 50, with respect to 1,000 simulation steps, this would then correspond to an absolute reduction in genomic replication, and very shortly after the initial time of infection. Now, after having defined our two model inputs, let's now start a conditional statement to define a block of code that's only executed when the simulation step is our time of drug therapy application. And what we would like to do is say that when the simulation time is equal to our variable time reduce, our time of drug therapy application, and the simulation time is equal to the drug therapy application time, then do the application of our reduction coefficient. Well, we can do this in Python. We're beginning with a conditional statement, if. And now we can reference the simulation time as passed by the simulator. You'll see this at line 124. The second argument for step is MCS. This is the value of the simulation step. We can say that if that value, MCS, is equal to our time of application, we do this by doing a double equal sign in Python. This is not an assignment equal, this is a Boolean equal to, if MCS double equal sign, time reduce. And then we can complete this conditional with a colon. So if the simulation time is equal to the time of application of the drug therapy, let's return. Twedit should add an extra level of indentation. This is necessary for Python. Anything that has this level of indentation will be in this conditional block. So if the simulation time is equal to the time of application, now let's do a loop over all of the living epithelial cells. So the immune cells don't have a replication model, and neither do the dead cells. But our other three epithelial cell types, the infected cells, the uninfected cells, and the virus-releasing cells, those all have active viral replication models, and we can modify the replicating rate by accessing the viral replication model per cell. Now let's do a loop over those three cell types. Every cell that has one of those three cell types. We can do this quickly in Twedit using a built-in code snippet. You can find this code snippet in the menu bar along the top. The fourth menu item in the menu bar of Twedit will say CC3D Python. And the very last entry in the first section will say visit. When you hover over visit in CC3D Python, you should find a second available item that reads all cells of given type. And when we click that, we find that Twedit will then drop in a template into your code wherever your cursor was. So if you didn't have your cursor in the right place, you can just hit Control-Z, and that'll remove what was inserted, and then place your cursor back appropriately where we would like to insert this line of code. Menu bar CC3D Python, down to the bottom to visit, and then all cells of given type. Now this is not actually an executable code. We try to also include some very basic instructions so that these code snippets can be filled out for your particular application. In this case, we can begin by taking the template code that was dropped in. Should be at around lines 137 and 138. We can just delete those two comments. And now at line 137, should have the beginning of a for loop that was very briefly mentioned in an earlier slide today. For cell in self.cellList by type. And again, this will do a for loop over every cell in simulation that has whatever cell types are passed as argument, as shown highlighted on my screen right now. And of course, we don't have cell types named type name 1 and type name 2, but we do have cell types named, say, uninfected. Again, uninfected, as with every other type name, by convention is capitalized in the stepables. So we can just pass the argument self.uninfected, where uninfected is all caps. We can pass another argument, self.infected, all caps. That is our second living epithelial cell type. And then lastly, our third type, the one that releases virus. In the model and in the documentation, we call it virus releasing. Currently, this has been refactored since then, but currently in the implementation, the name of this cell type is infected secreting. Now, when we pass the three arguments, self.uninfected, self.infected, and self.infected secreting, we pass this at line 137 to cell list by type. We'll then get an iterator. So we'll iterate over every cell that has one of those three types. And this is exactly what we would like to do. We would like to visit every cell that has one of those three types, and then we would like to refer back to the slides. Every one of those three cell types that has an active viral replication model, we would like to get the baseline parameter for the replicating rate, our max, and then we would like to apply our reduction coefficient. Now we can do this inside of our for loop as follows. For our cell of interest, we first like to access all of the available SBML solvers. We do this by typing cell.sbml. Now this will have an attribute for the viral replication model. The attribute is viral replication, where the R in replication is capitalized. Viral replication, the capital R for replication. And now let's start some brackets. Again, cell.sbml.viral replication is the instance of the viral replication model for the cell that we are looping at. And in the brackets, let's start a string. And to get the replicating rate for this cell's viral replication model, the string name is replicating underscore rate. Cell.sbml.viral replication with a bracket, and then replicating rate as a string. So that is the Rmax coefficient for this particular cell. And again, at our time of drug intervention, we simply want to reduce that coefficient for each of these cells that we're visiting. We want to reduce it by multiplying it by our coefficient defined by the variable magReduce at line 134. And so we'll do this in Python. This is as easy as typing asterisk equals magReduce. This is the same as saying that the replicating rate is equal to the replicating rate times magReduce. And that's it. So if your code looks like mine, then we are ready to test. So two coefficients, magReduce and time reduce, conditional block if the time is equal to our time of reduction, a loop over the uninfected, infected, and infected secreting cells, and then applying magReduce to the replicating rate of each viral replication model. Now, let's start first by testing to see if it works like it's supposed to. Now, if you have your coefficients, your model inputs at lines 134 and 135 different from mine, then for now, let's start by setting them to what mine are set to. 134, we set magReduce to equal 0. And at line 135, let's set time reduce equal to 50. Now, let's migrate to viral infection VTM model inputs.py. Along the top, viral infection VTM model inputs.py. And let's go up to the top and to line 14. This particular variable is another setting that's according to this framework, which is to plot the viral replication model of the initially infected cell. So you may have noticed if a graphics window didn't pop up, like it didn't for mine, plotting something like a viral replication model was because line 14 was equal to 0. So let's set the variable at line 14, plot VRM data freq for plot viral replication model data frequency. Let's set that equal to 1. Now what we would like to do is plot the viral replication model inside the initially infected cell, and we would like to find that at our very early application of the drug therapy, again currently defined as 50 simulation steps, we would like to find that at simulation step 50 in that initially infected cell, that there is a major noticeable change in the viral replication model, specifically due to the effects of having multiplied the replicating rate, the coefficient denoted as Rmax in the equations, specifically due to having multiplied that coefficient at simulation step 50 by 0. Now, let's make sure that everything is saved. So in Windows, I'm hitting Control S, making sure that I have no red disks along the top in Twedit. And once we have all blue disks in Twedit along the top, let's go to player. We have a simulation running in player currently. And you go to the top left and hit stop. And let's begin by clicking step. In player, stop the simulation if there was one running. And then we're going to click the second button, step. And see if we have any errors. If there are any errors in our Python script, they'll be reported in the console. Probably show up in red, and you had no graphics window screens pop up. Give everyone a second to diagnose any of those potential issues. Now, if there weren't any issues, then what you should find then is a new graphics window has popped up. You can refer to mine to see which one specifically. The window title will read VRM. This is for viral replication model. And referring to the slides, the 4 equations define rates for variables U, R, P, and A. And then back in player, in our graphics window for VRM, you'll notice that there are data points for U, R, P, and A. There are also plots for the variables uptake and secretion. Again, these are connections to the internalization and release modules, respectively. They don't show up very much with respect to the rest of the variables, so those won't be particularly interesting. What we'll be particularly interested in is the replicating rate. The variable R will show, hopefully, if we've implemented everything correctly, we should find, referring back to the slides, that at simulation step 50, when we multiply in our drug therapy, when we multiply this coefficient R max by 0, that we'll see a sudden change in the behavior of the variable R in our initially infected cell, which again is what's being plotted in the graphics window reading VRM. Now, before we start this, something else that we'll notice is that before this time, we'll also see some abrupt changes somewhere around simulation step 30. And what we'll find is when we refer to the epithelial sheet currently, we see lots of blue uninfected cells and one initially green infected cell, the one that's whose data is being plotted in the VRM graphics window. And when that green cell changes to red, this is the moment when the infected cell then becomes virus releasing, in which case some virus will start being removed from the cell's viral replication model via the secretion turn. And that virus that's removed from that cell will then begin to pop up in the virus field. So we should see 2 changes in the viral replication model. Let's run it. Let's go to the top left and hit run, the first blue button in the top left. And we'll see our first change due to the transition to virus releasing. So this is the eclipse phase. I see a drop in the variable A. And then at simulation step 50, we should see R start. Yep. You refer to my screen if you haven't seen it yet, or you can just wait and watch on yours. Should see at simulation step 50, our drug therapy has been applied. Genomic replication has been completely disabled. and all of the viral replication model variables go to 0. Now, if you got something that looks similar to my screen, then everything has been implemented correctly. And if so, then we can proceed back to Twedit. And we'll go to viral infection VTM steppables.py. And again, our simulations are defined for 1,000 simulation steps. Let's change our variable time reduce at 135 to 500. So this is applying our drug therapy halfway through the simulation. And let's change our coefficient at line 134. For mag reduce, let's change it to 0.75. We're going to apply a 25% reduction in the replicating rate halfway through simulation. Now let's save. Make sure we have blue disks all along the top. And we can go back to player. If anything is running, you can click stop. In the top left, the fourth blue button, stop. And let's just hit run. Now, if you still have your viral replication model data plotting, you should see there's the transition to virus releasing at around 22 steps or so. You should find now that at simulation step 50, there is no longer an abrupt change in the viral replication model behavior. Rather, you should see in particular that the variable R should be skyrocketing. This is again genomic replication. You should also see the variables P and A going up, unless of course your cell has died by now. At some point, this cell will be killed, either by virally induced apoptosis or one of the cytotoxic killing mechanisms by the immune cells, in which case you'll find that for your plot of the viral replication model data, all of the values will go to 0. Now, this drug therapy simulation was one that we found to be quite variable among simulation replicas. And so you may find as this is running, I'm at around simulation step 200. You may have some black screens popping up from your graphics window like I've been experiencing. Again, you can just delete those, create a new one, replace it, and hit play again. What you may find is that as you're watching your results play out, and then you flip back over to my shared screen and look at mine, you may find that you're getting something majorly different than what I'm getting. And this was precisely the intent. In my case, I'm at around simulation step 300. I've experienced lots of cytotoxic killing by oxidative burst, seen lots of virus-releasing cells get killed, but a nice layer still of infected cells. And the question is, at simulation step 500, depending on what all occurs leading up to that step, when the drug therapy is applied, the drug therapy in our simulation will either be effective to prevent widespread infection, or it won't. And that seems to be very highly dependent on both the spatial and stochastic nature of the complexity that's being simulated. So mine just passed 400 simulation steps. So mine is coming up on the drug therapy. My initial cell has been dead for a while, so my viral replication model is plotting 0 and has been for some time now. I would suspect that in my case, my simulation replica is pretty well advanced in infection, so I would bet that by simulation 500, that my replica is too far gone, that I'll see widespread infection where no uninfected cells remain by the end of simulation. Now my drug therapy was just applied, and yeah, mine is too far gone. So if you saw something different, that's interesting. Of course, feel free to leave that in the chat. It's interesting to see all of this variability for scenarios like this. I'll give it another minute to let everyone else's run for a moment more, and then we'll move on. Watching my population data, my last uninfected cell is a goner. Okay, so let's go ahead and stop our player. And let's return to the slides. Let's start on building our first stepable. Our first new stepable, that is. We can actually, very briefly, let's go back to Twedit. We don't need to delete the drug therapy code that we've implemented here. The easiest way to just disable this and leave it in the code for later, if you'd like to continue playing with it, at line 134, just set mag reduce equal to 1. And set time reduce at line 135 equal to -1. The first simulation step is equal to 0 and then increments. And so MCS never equals -1, in which case this block of code is never executed. So we can just leave that there. Now, one of the things that's not currently in the simulation framework is a model of recovery. So our simulations assign 20 minutes per simulation step, in which case we simulate just under 2 weeks of time. Now, referring to the schematic in the top right, this is a sort of aggregate schematic of a couple of different series of events for different pathogens. In general, we can expect that these sorts of behaviors will occur in some similar sort of transient behavior. And one of the things you'll notice here at the end of the schematic at around 14 days, which is just after our simulations stop, is this purple curve for accumulated tissue damage. And by damage in a simulation, as shown in the bottom left, damage in simulation is dead cells. So in this little excerpt from some simulation results, this is a nice little hole of dead cells. So this will be some damage in an otherwise uninfected tissue patch. Now, since our simulations haven't really considered beyond 2 weeks, we've not yet started to build things modeling recovery. And that's shown here with the tissue damage beginning to go down. Now, one of the complications with doing this in our current framework, and we're going to mess with this a little bit today, we have to consider the fact that for now, our epithelial cells in the framework are frozen. We've seen from the previous drug therapy scenario and in all of our simulations from the first workshop that the immune cells clearly move around, whereas the epithelial cells don't move. Now, clearly recovery occurs by proliferation. The cells that were killed are phagocytosed, they are removed, and then subsequently replaced. We can't do that currently without unfreezing the cells. CC3D certainly has lots of simulation functionality to be able to grow individual cells that are proliferative, and then to simulate mitosis and the introduction of new cells due to cell division. We're not going to do that today. Instead, what we are going to do is do some tricks based on our 4 different epithelial cell types. So the 3 are shown here: blue uninfected, green infected, and black dead cells. And our end result of recovery should be that those dead cells are somehow replaced with new uninfected cells. For the very simplest case, what we will say for this simple base model is that recovery is equal to a dead cell becoming an uninfected cell. So this is, depending on how this is cast, this is something like an epithelial cell underneath our tissue patch proliferating and replacing the dead cell that was there. We'll do a little bit more with this in the subsequent model development after this one. For now, we will do as follows. We will say that a model input for a simple recovery model represents the time that it takes for a dead cell to be replaced with a new living uninfected cell. So our one model input will be a representation of how long it takes on average to replace a dead cell with an uninfected cell, and we will do this by assigning a probability of recovery. And this probability of recovery will be assigned for each dead cell at each simulation step. In which case our algorithm will be, for every simulation step, we'll then do a loop over each dead cell in simulation, and we will consider the probability of that dead cell being changed to an uninfected cell. And yeah, let's do it. So let's go back to Twedit. Now, of course, you can go through and begin by manually typing your own new steppable. Twedit provides some functionality to streamline this process. You can do this by going to the project pane on the left and selecting viral infection VTM steppables.py. If your viral infection VTM.cc3D is not showing these simulation scripts, you can expand it by clicking on the little black icon next to the CC3D icon. That should show viral infection VTM steppables.py. And you can right-click that item. Right-clicking that item will present a drop-down menu item that says Add Steppable. So we right-click viral infection VTM steppables.py, go to the third drop-down menu, Add Steppable, and click it. Now this should open a new dialog window. that reads generate steppable. Now we can leave all of the default settings as is and just give our steppable a name. Let's call this steppable recovery steppable. One word, capital for recovery, capital R, a capital S for steppable, recovery steppable. So if your screen looks like mine, then you can click OK. And this should take you to the bottom of viral infection VTM steppables.py. You should find at around line 1071 that a new steppable has been created by Twedit for us with our familiar steppable functions init, start, step, and one that we won't use today, which is finish. Now, one thing you'll notice at line 1071, Twedit automatically assigns inheritance for our new steppable from the CC3D Python steppable base class. You can see this in line 170 or 1071 steppable base pi. We would like to replace that with the base steppable that's been defined for this project. And we can do that by just scrolling up to line 990 to oxidation agent model steppable. At the beginning of the definition of that class, you'll see that this inherits from viral infection VTM steppable base pi. At line 990, you'll find viral infection VTM steppable base pi in parentheses. You can just highlight viral infection VTM steppable base pi and copy. And then we'll go back down to our new steppable at line 1071 and replace steppable base pi by pasting viral infection VTM steppable base pi. After having done that, we'll then go down to line 1073. You see another usage of steppable base pi. Line 1073, let's highlight steppable base pi and paste again and replace it with viral infection VTM steppable base pi. And so now our new recovery steppable will not only have all of the built-in attributes and functions and so on and so forth defined with the normal steppable base pi, will also inherit all of the additional features that we've developed for the viral infection modeling project. Now, for our recovery steppable, we won't need to do any initializations, so we can just delete our start function. So you can begin highlighting from the end of line 1078, highlight all the way up to the end of line 1073 as I've done on my screen. You can just delete all of that. And you can delete that such that the step function should start at around 1075. Now, Twedit also adds a couple of little template examples into every auto-generated steppable. You'll find this starting at around line 1076. You should see a print statement printing something about recovery steppable and then a for loop over the cell list. You can just remove all of that as well. Let's start with a blank step function. Now, in our empty step function, let's start by defining a new model input for our probability of recovery. And we're assigning a probability for each dead cell at each simulation step of becoming an uninfected cell. And we'll do this at the beginning of our step function. Let's call this new variable recovery underscore prob. Recovery underscore prob. And let's say that on average, this should take about 7 days. Now we can make this approximation by doing the following. We have a model input that defines the physical time that's assigned to each simulation step. And that variable is called S underscore 2 underscore MCS. So let's start by saying that our recovery probability is equal to S underscore 2 underscore MCS, as I've done in mine. Now this is defined in seconds. Let's divide that by 7 days in seconds. So divided by, we'll start a set of parentheses. Then we'll say 7 days times 24 hours per day times 60 minutes per hour times 60 seconds per minute. So our recovery probability is equal to S to MCS divided by 7 days in seconds. Now let's start a new block of code. We would like to do a loop over every dead cell at every simulation step. So this is a loop over cells by type again. And we can quickly generate this using a code snippet in Twedit by going to the menu bar and going to CC3D Python. And to the bottom, go to Visit. And then the second item, all cells of given type. This will insert a template with this type of loop. Again, if you didn't have your cursor in a place where you would like Twedit to enter that, you can just undo and place your cursor in the right place, and then Twedit will insert the code there. And there's some basic instructions for usage on this. We can delete the comments that were inserted. Should be around lines 1078 and 1079. Just delete those comments. You can delete the comment and print statement inside of the for loop. And now let's fill out our arguments to self.celllist by type. We're going to do a loop over all dead cells. In this case, we'll start by passing one argument, which is self dot, and the type name for the simulation framework that represents dying or dead cells is dying. So self.dying, where dying is in all caps. The dying type are dead cells. And we'll complete that with a colon at the end. When you hit return, should find Twedit, add a new level of indentation. And now let's test for recovery. I'm going to return to the slides very quickly. I'm going to draw a uniformly distributed random number in 0 and 1, and then see if that random number is less than our recovery probability. If the uniformly distributed random number is less than our recovery probability, then we implement recovery. So let's go back to Twedit and do this. Should be around line 1079. Let's start an if. Now let's draw a random number. You can do this by entering NP. This is NumPy being imported as NP. NP dot random dot rand parentheses. If np dot random dot rand parentheses. So this is our random number, and we'll say that if our random number is less than recovery prob, our recovery probability. The random number is less than the recovery prob. Let's finish the conditional statement with a colon and then hit return. But it should add a new level of indentation. So in the case that the random number is less than the recovery probability, recovery occurs. Let's start by changing the cell type to uninfected. This is a dead cell experiencing recovery. We can change the cell type to uninfected by entering cell.type is equal to, so self.type equals, or cell.type equals self.uninfected, all caps. Cell.type equals self.uninfected, where uninfected is in all caps. Now, ordinarily in CompuCell, you can just change the cell type as we've done, and that cell type change is implemented. For the simulation framework, there are a couple of processes that are associated with initializing modules that go along with living cells. These are processes that are disabled when a cell type dies. Now what we would like to do is reinitialize those processes. Now we don't have to type all of them out explicitly. Instead, we can jump to the top of viral infection VTM stepables.py, and we'll scroll to line 58. So in viral infection VTM stepables.py, line 58. These are some processes to just initialize the viral replication model in each cell, reset the viral replication model variables. We model a dynamic number of surface receptors in the internalization module. Those are initialized at line 60. And then beginning at line 61, this is just loading the viral replication model for a cell. Now, all of these need reset when we introduce our uninfected cell back into the simulation. And we can just copy these 4 lines. Let's highlight everything from line 58 to line 61. And let's copy that. So everything in lines 58 through 61. Please note that we're only copying the first 2 arguments to the function load viral replication model. We don't need the rest. So copying lines 58 through 61, let's jump back down to our new recovery steppable. And let's go to, it should be around line 1081, 1082. And let's just paste there what we just copied. So you should have 4 lines. Begins with cell.dict with a key equals false. Quick question from Christian about what the S to MCS contains. S to MCS is a model input that describes the number of, or the amount of time per simulation step. In this case, for our simulations, each simulation step is equal to 20 minutes, in which case S to MCS is 20 minutes in seconds. So from lines around 1082 to 1085 should have 4 lines of code that were copied. The last of those 4 from our copy needs a little bit of completion. So we should have the arguments cell equals cell and VR step size equals VR step size. Should be the only 2 arguments being passed to low viral replication model. You may have a comma after VR step size. Let's replace that comma with a parenthesis that completes the parentheses for the function low viral replication model. And that's it. So if your 4 lines of code look like mine, then this is all set up. Now let's jump back to the slides very quickly. So what we want to do is test this to see that this is doing what we think it should. Let's generate something that looks like the sample results in the top right of this slide. What we will do is we'll generate a test case by implementing just a couple of additional lines. What we'll do is that we'll start a simulation, and we will say that at 200 simulation steps, at step 200, we're just going to turn off internalization. I'm going to say at line 200, something happens that disables internalization. And the reason that we'll do this is so that we will allow some infection to propagate up to 200 steps. And then what we want is to see that infection get contained and eliminated to generate a patch of damaged tissue and then we can watch recovery happen. So we can do this using this command shown here. This is using the shared steppable vars, and this is a global dictionary that's accessible to all steppables. This is where a coefficient in the internalization model is defined. This is such that when we set this parameter to a very small value, internalization is effectively disabled. So we can move over to the code. Let's implement this little test very quickly. We can return down from the end of our for loop. Let's reduce our indentation back to the same level as the for loop that we just defined. When we reduce that level of indentation, that gets us out of the for loop in Python. And now let's set up a conditional to execute a block of code only at simulation step 200. We can do this by saying if MCS, it's the current simulation step, equal to the double equal sign, MCS double equal sign 200 colon, the simulation step is equal to 200, and let's disable internalization. Self dot shared underscore steppable underscore vars, and then a set of brackets, start a string, and then k underscore on. So this is the coefficient in the internalization module that we're going to modify to generate containment of infection, and a damaged tissue patch. Self.shared steppable vars, the keyword or the key of K on, say equals 1 E negative 12. We can't set this to 0 because it'll blow up the simulation by a division by 0 error. But we can set this to a really small number. This will effectively shut off the internalization module. This will generate our damaged patch. Now, if that's implemented as it looks on my screen, then let's go over to player. Let's hit stop if anything is running. And let's hit play. Now what we should see as we look at our epithelial sheet, we should find that for the first 200 simulation steps, that infection progresses as we've observed it in lots of different cases. See some initial virus release by the first infected cell, start seeing neighbors become infected very soon. And then at simulation step 200 in our test, we should see no more uninfected cells become infected. In which case, everything after that time that's been infected will die through various killing mechanisms. And this will generate our dead, damaged tissue patch. And then we want to watch that area where all of the dead cells can be found and see if we notice some of those dead cells becoming uninfected. So mine's at simulation step 180, coming up on 200. I already have some death. My internalization module has now been disabled. I should see very little okay, so mine didn't work, and the reason it didn't was because I didn't save. So one thing you'll notice when going back to Twedit is that inserting a steppable using the features in Twedit will actually make changes in 2 different scripts. And if you refer to my screen, you'll notice that I was doing work in viral infection VTM steppables.py. I had saved. And I have my blue disk for this Python script, but I have one that's not been saved. I have a red disk for viral infection VTM.py. And in this particular simulation script, this is how CompuCell is told about your steppables. Notice most of these steppables should look familiar. SimData steppable, looking in viral replication steppable earlier. And Twedit went ahead and informed CompuCell in this script of recovery steppable, but I hadn't saved. So in this script, I'm also going to save now. Now I get a blue disk everywhere. Switch back over to player. Simulation has been stopped. I'm going to restart by hitting run. I'm going to restart this simulation. Watch this, look for our new effects from recovery steppable, and then we'll close out for the day. So I'm at simulation step 50. I have a bit of spread of infection now. Got 1 infected cell, initially infected cells, now virus releasing. coming up on 200 steps, at 130. Spread of infection, 200 simulation steps. Expect to see spread of infection stop due to our disabling of the internalization module. We'll then watch everything die, get a nice patch of dead cells, and then we can observe what recovery steppable does. I'm at 220 steps, don't really see much more spread of infection. See the remaining infected cells become virus releasing, see some cytotoxic killing. These new like sudden bursts of patches of dead cells. This is from secretion of the oxidative agent by activated immune cells. Coming up on 300 simulation steps, just about everything is dead that was infected. My dead patch kind of looks like Pac-Man. And if your replica is about to where mine is, then you may notice that there are some uninfected cells popping up at random inside of the patch of dead cells. And some of them will become uninfected and then suddenly go dead again. And this is due to the amount of oxidative agent that's still in the area. So an uninfected cell is being introduced and then getting killed again. Now my replica is coming up on 400 simulation steps, and now I see uninfected cells being introduced into the patch of damage, mostly random. This is clearly filling in. So this is a very basic elementary simulation of these dead cells being replaced by some imaginary available epithelial cells underneath. You see this here. Presumably by the end of simulation, this will then fill into an undamaged patch. So we have about 4 minutes left on the workshop. We've prepared a couple of other demos to get through today, just seeing how time goes. Obviously, we don't have time to get to those today, but again, in the Google Drive shared folder that we dropped into the chat, we'll drop that in again. In that shared Google Drive, you can also find all of the simulation files with those different demos in their completed state. Also, these slides are available so you can go through and see what the basic premise of those demos are. Here is the link to the Google Drive again in the chat. As we've mentioned a couple of times, we are very eager to collaborate to continue developing this framework. As you can see, there's a lot to be done with it still. There's a lot of complexity associated with viral replication and infection that spans from the subcellular all the way to the whole body scale. So we're very eager to work with both modelers and experimentalists alike. We do like to do lots of cross-platform validation. So we work with friends who develop Morpheus and Chase, implementing versions of the framework in their platforms as well. Very grateful for the funding that's provided to us to be able to do this work, both from NIH and NSF. We also have quite a few collaborators from a lot of different backgrounds who've been involved in developing this framework, and we're extremely grateful to them for their participation, all the insights that they provide. If you'd like to know more about getting into the basics of CompuCell3D development, we hold a summer workshop every year that begins at basic Python programming language and goes all the way through to individual and team-based hackathon-like work. So introduction to Python all the way to advanced model building. So you can find more information on that at our website. It's CompuCell3D.org. With that being said, this is our contact information for Professor Glazier and myself, shown here at the top right. Again, please feel free to contact us for support issues with local installations, for any issues you may have had getting through today, any interest, whether it be through modeling different aspects of the complexity of viral infection, doing experimental work, so on and so forth, getting into doing the demos that we weren't able to get to today, anything in particular of interest, please feel free to reach out. You can follow and fork the project on GitHub using the link below. We'll include that link in the chat again very quickly for your reference, so you can stay up to date with development of the project there. Again, everything is open source and we update this as we develop it. With that in mind, we are just at noon o'clock here in Indiana. This concludes our time. Have maybe a little bit of time to take any final questions or comments, or James, if you have anything that you would like to include to close us out. I'd like to turn this over and stop talking now.
Okay, I'd just like to thank you all for joining and say that we look forward to hearing from you and supporting you if you choose to use this tool. And it doesn't have to be on viral modeling. CompuCell is usable for all sorts of multi-scale biological simulations, and we'd be happy to talk to you about other possible applications as well. Thank you again for taking 3 hours of your time today, and we look forward to working with you in the future.
Thank you very much, everyone. Have a great day.